And I'm happy to introduce Adam Butterworth, Hauke Haukenerson, and Gadi Rennert, who will be moderating this session, and they'll handle questions, and I'll just sit back. So Adam, Hauke, and Gadi, take it away.
Sure, thanks, Terry. Well, welcome back. It was a great session we had earlier with fantastic talks, and I'm confident, given the exciting speakers we have for you in the next session, that we can continue the high quality. We heard yesterday in the, those of you who could join the Scientific Strategy and Cohort Enhancements Working Group session, some of the initial work that's come out of the pilot project on polygenic risk scores, and we also touched on some of the other ideas for pilot projects going forward, and we discussed the opportunities that are going to arise after this meeting for kicking off some of these other pilot projects. So we really wanted to bring together some speakers who could give us insights and challenge us about ideas that we could take forward. So we're excited to hear from all of the speakers in this session. So without further ado, let's make a start. The first speaker is Dr. Alicia Martin. She's an instructor in investigation, as you see on the slide here, at Massachusetts General Hospital, and also a member of the Broad Institute. And Dr. Martin's really pioneered a lot of the work that's gone on looking at genetic studies of diverse populations, including polygenic scores, so perfectly aligned with some of the work that's going on in IHCC. So welcome, Dr. Martin, and very interested to hear your talk. And before you start, maybe you could confirm where your exciting background is for us.
Sure. So you don't have to look at the rest of my apartment. My exciting background is in Cape Town, looking out from Table Mountain. So nice view sweeping into the water.
Fantastic. Please go ahead.
Okay, great. So my presentation is going to be mostly about how we can start to think about facilitating diverse population genomic studies and about all of the advantages that I see this getting us. As you all are very much aware, our genetic studies are becoming increasingly powerful. Over the course of the last decade or so, our cohorts have grown from the scale of something like hundreds to maybe thousands of individuals to now routinely having participants on the scale of millions of individuals in some very large scale efforts. Unfortunately, however, this has not happened in an equitable manner necessarily if we're looking at human genetic diversity. So now about 80% of participants in these large scale genetic studies are of European descent, which is far out of step with the global population, where European ancestry populations make up about 16% of the global population. Additionally, this is really problematic if we think about diversifying progress. If you look at how the non-European representation is tracking as a function of time, you can see that the non-European representation in these GWAS peaked around 2014, and since then has stalled or perhaps even slid in the more European direction. This is clearly a problem for potential global equity if we try to think about how these large-scale genetic studies are going to transfer to other populations. So that's really a key question that I've been working on, is trying to understand how these large-scale genetic studies and the information that we're learning from them are really going to transfer to other populations. So the tool that we use to try to investigate this transferability is a polygenic risk score. So I'm sure you've all heard of this at this point in time, but a polygenic risk score is simply the genetic prediction of an individual's phenotype. So we take a large-scale genetic study that's been done before, we multiply the effect sizes that come out of that by the genotypes that are present in a target cohort. So in this way, we can understand how much of the information from genetic studies is transferring to different populations. Additionally, we can use this in all sorts of different contexts. In thinking about deeply phenotyped cohorts and starting to think about clinical translation, preventative medicine. Those areas I think still need a little bit of fleshing out, but it's a really exciting and promising area. So the biggest potential issue then with thinking about something like clinical translation are the massive disparities in polygenic risk score accuracy. So last year we looked at polygenic risk score accuracy in the UK Biobank. We used a European discovery cohort, and then we held out some European ancestry individuals and then also looked at the non-European ancestry individuals in the UK Biobank to try to understand how predictive these polygenic scores are in a globally diverse cohort. And the disparities in accuracy are really staggering here. So if we normalize with respect to how we're doing in European ancestry populations, you can see that we're doing about twice as well in these European ancestry populations as we're doing in East Asians, and we're doing about 4 to 5-fold as well as we're doing in African ancestry populations. These are really big differences, and we understand some of the genetic basis here. So when we're constructing these polygenic scores, these predictors, we're building these from correlated variants, not necessarily causal effects. So even if the causal effects are exactly the same, we're not necessarily building predictors that are equitable. Additionally, the reason that these may not be equitable also can boil down to the fact that there are differences in allele frequency and LD here. So we really need a comprehensive framework, I think, for facilitating these diverse studies. And this is not going to happen by pushing in any one area. We're going to need to have sort of a systemic effort in pushing on developing new methods to analyze globally diverse populations, developing new data and community resources so that we can reach these underrepresented populations and serve the genomic infrastructure that we need to sort of improve data quality there. And then we also need to increase research capacity, train the next generation of researchers to lead these efforts in their home countries to ensure the sustainability of these efforts. And with this is going to buy us a lot of really exciting scientific innovation, including things like being able to find map variants better, leveraging the LD differences within and across populations to more accurately pinpoint causal variants. This will also identify novel associations that are polymorphic in non-European ancestry populations, and that are at low frequency in European ancestry populations. So this will identify some low-hanging fruit in these groups. Additionally, we can use this to uniquely inform those shared versus unique genetic and environmental risk factors and the determinants of health in these different populations. And then an area that I'm particularly interested in, keen on working on, are genetic risk prediction differences. So we can improve accuracy for everyone, especially in underrepresented populations. I'll start to show you a little bit of pilot data there. So just on the method space, this is just an example of one area that we're pushing on. So this method that we're developing is called multi-ancestry meta-analysis, or MAMA as we call it for short. This assumes you have GWAS from 2 or more different populations. And this is unique in that we consider both within ancestry LD, but also cross ancestry LD to recalibrate effect size estimates for each population. This works as an extension of LD score regression and MTAG. And we show that this is an unbiased method that improves power compared to other unbiased meta-analysis methods that are designed for multi-ethnic purposes. This is a really collaborative effort, and I'm really lucky to be working with these folks on this. So in terms of the data and resources front, one of the big gaps in cohorts that we have across the world is looking in Africa. So one area that I'm really excited about is this development of the neurogap psychosis cohort that Keristin Conan is really driving. So this research effort is on track to study about 35,000 Africans spanning Ethiopia, Kenya, Uganda, and South Africa to learn about the genetic risk of psychosis. This is going to be the largest case control cohort that's ever taken place in Africa. So of course, you can imagine how much infrastructure building there is to go along with this. This includes things like building genomic resources for African genetic studies and making sequencing versus genotyping recommendations. This type of work is also not feasible without a very rigorous and in-depth training program. So we have an on-the-ground program called Ginger that I'll mention a little bit more, and also ensuring that participants on the ground get study resources and something back instead of having some sort of safari research go forth, take samples, and never get back to the local community. So in terms of the research capacity element, which I think is really, really critical, this has been a huge investment led by Lori Chibnik, which I think sort of can act as a model for how we think about ensuring the sustainability of this type of work. So this has so far been a 2.5-year training program that is very immersive, involves immersive workshops, online weekly classrooms, and on-site trainings catered to the needs of local institutions. I'm showing 3 of the research fellows that I've been mentoring on an in-depth research project. So Nastasia Larato and Alan have really been leading some research looking at the generalizability of polygenic scores to different African populations. So just to mention and shout out to them a little bit more of this work, they've each been leading 3 different elements of this project, but one is really looking at testing the transferability of polygenic risk scores across diverse African populations. For example, extending the work forward that I mentioned before to look within Africa. Africa is an enormously diverse continent, and so it's, I think, really important that we look at the diversity present there to try to understand how these risk predictors are going to transfer across different populations. So you can see that there's quite a bit of difference across these different populations, across recently admixed West African and British individuals, Ethiopians, East Africans, West Africans, and South Africans. Uniformly across the board, we're doing worse at predicting in Africa than we are anywhere else. But that's not to say there's no promising efforts here. Sorry, the figure is a little funny here. So if we look at how we're doing in terms of predicting different traits, you can see, for example, if we look on the left parts of each of these coupled plots, that shows how we're doing in predicting different traits with respect to European ancestry populations using only the UK Biobank Discovery Cohort. And on the right side, I'm showing you how we're doing at predicting these different traits in these different populations when we use UK Biobank coupled with the Biobank Japan, as well as the PAGE study, which is the population architecture and genetic epidemiology study, including a lot of African Americans and Hispanic and Latino populations. And so you can see that there's disproportionate promise from including these non-European cohorts, and that we're particularly gaining in recently admixed American populations, Hispanic and Latinos, East Asians, and in African ancestry populations. There's some notable outliers in these African ancestry populations. These come from things like mean corpuscular hemoglobin concentration and white blood cell count. There are some really large effect variants that are present, for example, in African populations that are not present in European ancestry populations. Again, driving home the fact that we're going to be discovering novel low-hanging fruit in these underrepresented populations that we wouldn't be able to discover with a European-only study. And these are also going to help us drive forward and improve the prediction accuracy in these populations. We should also be making full use of the biobanks that we already have collected and to hand. So I've been leading an effort to analyze all of the non-European ancestry participants in the UK Biobank. So here we know that, for example, we have about 420,000 individuals of European descent, not all of whom we're making use of in most current analyses due to relatedness and more population structure and those sorts of things. There are also, notably, however, also very large sample sizes of Central and South Asians on the scale of about 9,000 once we do some pruning for outliers. African ancestry participants make up about 6,700. East Asians, we've got about 2,700. Middle Easterners, 1,600. And admixed American populations like Hispanic and Latinos make up about 1,000 or so. We've now run GWAS in a linear mixed model framework, accounting for population structure in thousands of phenotypes. So that right column in that table shows you how many phenotypes we've performed an association analysis for, and for different continuous traits, for different biomarkers, ICD codes, prescriptions, Phe codes, which are aggregations of these ICD codes, and categorical variables. This is, of course, a very collaborative effort that I'm working on, for example, with Hilary Finucane's lab, Ben Neal's lab, and other folks. So this is going to be very exciting. We're planning on releasing these summary statistics out to the community soon, including meta-analyses across these different populations, such that people can very easily make use of these data. And just say thank you to the breadth of collaborators here who I'm really excited to have been able to work with on these. Thanks.
Thanks very much, Alicia. Fantastic talk, and so relevant to the work that's going on in IHCC and the work we want to do. Fantastic. A couple of messages, questions I saw coming in the chat. So Jeff Ginsberg, you asked about, could you comment on how you standardize the phenotype measures across these diverse populations so that we're sure the genetics is reporting on the same thing?
Yeah, that's a good point. We usually try to make use of data-driven approaches through looking at things like genetic correlation across populations. Of course, a challenge there comes to the differences in LD. So there are some methods that have been developed in this space, things like popcorn, for example. It's also really important that you look at just like the standard distributions, like you look at the distributions of the phenotypes across these different populations, you look at the prevalence differences, all of those sorts of things. So really we are doing analyses primarily from traits that we look at that show evidence of strong genetic correlation across populations, and their phenotypic distributions look the same.
Great, and Jeff comments that the PhenX presentation later might be useful, relevant to that as well. One more question then from Lucia Hindorf, who says, what's your take on how far we can get in terms of improving polygenic risk score prediction in diverse populations using existing data versus needing to generate new data?
That's a great question. So I think right now, if we look at the sort of where we're at, we're doing a pretty poor job in African ancestry populations. We're going to do basically corresponding to genetic distance and population history. That's going to sort of bound our accuracy. So we're going to need to invest most heavily in those populations that are most genetically distant from European ancestry populations. This is just simply a function of human population history and human evolutionary history on the scale of, hundreds of thousands of years. So I think we're going to need to collect a lot of new data where we're doing the most poorly, and then we're going to need to integrate harmonized data where we already have access to it, in places where we're not doing quite as poorly. So I think we can make quite a bit of headway in the US, for example, in Hispanic and Latino populations, because they're less genetically distant from European ancestry populations than, for example, in African Americans, where we're going to need to really start to invest in those African ancestry populations to make any headway. But it's all sort of like a function of this diversity spectrum.
Great, and that's a great message for us to end on and to take forward into our deliberations about future IHCC projects. Thanks very much. I'm going to hand over now to Håkan and introduce the next speaker in this session.
Yeah, thank you, Adam. So the next presentation will actually be shared by Dr. Elias Zerhouni and George Vandenberg. And I'll introduce both of them now, and they will just decide who presents what. Dr. Zerhouni is currently a professor of radiology and biomedical engineering at Johns Hopkins University, where he spent most of his academic career. And among his many roles there, he served as the executive vice dean for several years, and he became the director of the NIH, as most of you know, in 2002 under the Bush administration. And he also served under the Obama administration, where he played a critical role in fostering scientific and technological collaborations with other nations. And most recently, he was the president for global R&D at Sanofi. And Mr. George Vandenberg is an attorney by background. He served as the chief counsel of America Online and senior executive of the AOL, Time Warner, and Fox Broadcasting. He's the chairman of and co-founder of the US Against Alzheimer's, which is USA2. And this is an Alzheimer advocacy organization committed to do what it takes to basically stop and cure Alzheimer's disease. And they will be presenting on behalf of the Davos Alzheimer Collaborative on the developing a global cohort for Alzheimer's disease. So this is really a great synergy with the IHCC, as we discussed briefly yesterday. So please, Elias, George, stage is yours.
I hope Elias is on, and because he was called into an emergency meeting at our HHS agency. So he may join in progress. So let me start, and then he will join if he can in breaking out of that meeting. So this is the Davos Alzheimer's Collaborative is a partnership of the Global CEO Initiative on Alzheimer's disease and the World Economic Forum. The Global CEO Initiative is a patient-led collaborative of the major pharmas and biotechs who have molecules in the Alzheimer's space. But it is patient-led. My own family, I've lost 2 generations to this disease, and I am in the midst of losing a 3rd generation, and that's over a 40-year period. Absent acceleration, my kids, my grandkids are at risk. We are going to propose a collaborative with you to use Alzheimer's as a use case for all of the extraordinary technology that you've heard about in genetics and phenotypical cohorts around the world. We want to actually break down the barriers between cohort-based research to discover loss of function, mutations, and other genetic risks and other risk factors for Alzheimer's in different populations so that we can make sure that the drugs that are developed not just with the idea of serving European-based populations, but the populations of the world. As all of you know, Alzheimer's or dementia, depending on how the country uses the term, is present in every country of the world. Now 50 million families, predictably going to 150 million families by mid-century, which is close to 500 million people. So we're talking about something that affects everybody. So we really want to deepen and diversify our cohort-based research to explore all pathways of greater global scope, engagement, and heterogeneity, and with the tools that you're already developing as a platform to use those new analytic tools in partnership with you as we design a scientific plan here. So the problem is we have an incomplete understanding of the heterogeneity of this disease because we have studied it largely in European-based populations. We need to expand that work into much more heterogeneous populations. and low and middle income countries to assure that we identify new targets and associated biomarkers. And we build a broad cohort to deepen our understanding of the genotypical, phenotypical, and biotypical diversity of the disease across the globe, and use the shared infrastructure that you're developing to understand that. So I think Elias has joined. Elias, you want to offer some remarks? Because I know you have a very limited time here, given your
very little. I think you're doing exactly a perfect job, and I don't have much to add except to say this: if you look worldwide, one of the components that is lacking in trying to understand and translate all the progress we've made at the fundamental level in Alzheimer's disease is a coordinated cohort that actually, very much like the other presentations we've had today, for example, on the UK Biobank, brings people together or able to build a coordinated cohort around new scientific plans. The idea that there are areas of progress that need to be made, and there are discoveries that we're making at NIH and in conjunction, for example, with the IAMP consortium. All of those will need to be tested and really validated. And we do understand that the disease is heterogeneous, the disease is global. We will win by collaborating and providing means to the cohorts here in IHCC to be able to study the subcomponents of the cohorts that they would be interested in pursuing. We're not interested here in just taking members from the cohorts and patients and then combining them. We really want this to be a collaborative work around, as you said, new ideas, new platforms, new technologies, and new ways of looking at Alzheimer's disease. Not unlike what we just heard about, loss of function mutations that are protective, discovering them and validating them. And we've set a process, which is on your slide number 5, which would basically convene a working group with partners, the partners who are interested. This slide essentially shows that there will be subcomponents of each one of our cohorts here that could be part of a more global effort and more integrated effort. I just looked at the biobank data that was presented. Altogether, there are 10,000 people who seem to have Alzheimer's disease in the total sample, if I'm correct. That is not enough. We need a larger number of patients that are fully biotyped, fully phenotyped, followed over time to understand the heterogeneity of the disease, and eventually find an accelerated pathway to discover new targets and implement efforts around them. But it would require coordination. It would require, for example, to come together for a scientific plan that will be exciting to all the cohort leaders, that will justify additional funding for all the cohorts and the central coordination of that with standard methods and standard ways of doing the phenotyping as well as the biotyping. We need to understand the landscape. It's clear that we do not have a grasp of the landscape, and I want to thank you for agreeing for us to speak and share with you what our ideas are. And from that, draft a scientific plan that hopefully will be bold enough to be of interest to all of you. And then once we finalize that, really work on something that George and I and others will work on, which is that we need funding for this. And we certainly are not asking for cohorts to contribute funding, but to contribute their knowledge, their wisdom, and their interest in this effort. So you can see this Davos Alzheimer's Collaborative is really based on 3 fundamental directions and goals. And I'll turn it over to George, who has been leading that conversation.
So you'll see here that the global cohort associated with identifying new targets is associated with, obviously, biomarker development, but it also is connected to an effort of the Davos Alzheimer's Collaborative to develop a unified global clinical trial support system so that sponsors are presented with a uniform experience in getting protocols adopted, a faster start to trial, faster completion of trial, and creating platform trials in the future as we know more about the disease. And then to prepare the healthcare system, which now does not detect this disease early, does not have clear clinical measures or preclinical methods of diagnosing the disease as we prepare the health system. So this is a development activity with you that will tie in directly to linking it to clinical trials and to the health system preparedness that we need globally in order to make sure that the innovative medicines that emerge from the new targets that come out of this cohort work will be delivered to patients worldwide. Great danger that we miss the rest of the world. So we do have a poll here, and I know this is fast and this is very summary, but we would like to know whether you're interested, because what we are going to do is actually have a call. We're going to have 2 calls to go deeper into this proposal and to discuss it with people who may be interested in doing so. So we very much would like to have at least an expression of interest and a follow-up call on one of 2 days, either May 19 with the times indicated or May 20 times indicated, basically to understand what we're proposing, how you might get involved, how the cohorts might participate in a co-delivery, co-development effort for building a scientific plan, informing a business plan, creating a 5-year budget, and the associated financing of that. So I think the poll is up now, and obviously anyone who would like to express interest, please do so. And through Jeff's good offices, we will connect those who express interest to connect on one of those 2 phone calls.
Yeah, thank you, George and Elias, for such an inspiring presentation. And this is obviously a great initiative and collaborative opportunity, and sort of the mission and vision here. Obviously, it's going to be transformative with respect to this disease if successful. Are there any questions from anyone? Maybe start by sort of asking how do you envision sort of the best sort of the initial interaction with the IHCC in terms of what should really be our first steps. As I mentioned yesterday, we are compiling information on dementia and Alzheimer's disease across the cohorts within the IHCC network, 97 cohorts total, over 50 million people. So we envision we will have a very large number of potential candidates. How do you think it would be best to sort of, move forward to engage these cohorts in this global initiative?
I think what we need is with the IHCC, namely you, Håkan, and a selected group of cohorts, to almost very quickly get a clear understanding of what our scientific aims are here and what the scientific plan of operations might be. You already have an enormous set of assets that can be turned to this. The polygenic risk scores, understanding all the genetics of the disease are rather critical, and they will be different in the heterogeneous populations represented by your cohorts. There are comorbidities associated with this disease, not just the familial connections that were mentioned earlier, but also cardiovascular disease and diabetes. Also, we know that lifestyle makes a difference, and probably makes a difference in different cultures differently. So you have an enormous amount of already assets that you're beginning to accumulate and understand that have a bearing on this. There are going to be some new assets that we're going to need, probably, which are going to be the ability to detect certain proteins in individuals. So that would be a set of biospecimens you may not have, and then we can't scan people, I don't think, at scale. So I do think, though, that there will be a blood-based marker that will be FDA and EMA approved by next year. So going to where the puck is, I think we can use the the blood and plasma samples that you have in order to detect the presence of underlying pathophysiology. And then, of course, we'll need some clinical measures. And my own judgment is that we will be developing, potentially with you, a digital-based distance and remote capacity to detect clinical variations. As one of the presentations earlier demonstrated, simply the speed at which you can process and utilize filling out forms may be a very good initial signal of the presence of some cognitive impairment. So I do think you have an enormous number of assets already, and I think we need to identify what we need to build on top of that in order to really get at the core problem so that we're confronting for Alzheimer's.
So I would just add, if I may, just one point, because I think George covered it very well. One of the things that is really important is to define a staging mechanism for the disease. And in addition to heterogeneity, progression of the disease is not uniform across patients. What would be very interesting is longitudinal data. Why is it that some patients evolve through the disease very quickly and go from stage 0 to stage 5? What are those stages? How do we characterize them? And why is there a difference between those who evolve slowly and those who evolve rapidly? Can we not discover protective or mechanism or gain of function, loss of function changes? And those are questions that I don't have the answer to, but I do know that there could be a convergence of scientific views there that will make some, if not all, cohort participation worthwhile.
Yeah. No, that's certainly it's a great opportunity. Now there are numerous questions on the chat platform. Maria De Silva just pointed out that there is a UK Dementias platform online. And we had similarly discussed the Alzheimer's genetics consortium here in the US. So, there are numerous of these sort of opportunities to better integrate and bring people together with that same mission and focus. Terry, do we have time for one or two more comments, or do we need to move on? There was a survey, as you know, that is going to address the interest of the PIs on these cohorts, as Georg pointed out. Great. Well, George and Elias, thank you so much. It was very, very...
Thank you. Thank you for having us.
Yes, thank you very much.
Thank you.
So we'll continue now to the last talk of the session. And Dr. Aaron Ramos from the NHGRI will present the PHENix toolkit consensus measures for phenotypes and exposures. Thank you.
Well, thank you so much for inviting me. I really appreciate the opportunity to provide this brief overview of the PHENix toolkit. Sorry if it looks like I'm staring at you deep into your eyes, but I need to get close to my laptop screen. So as the IHCC thinks about addressing new research questions, the PHENix toolkit may be a useful resource for you as you explore which data collection instruments that make the most sense given your particular questions you're exploring. So just a little bit about the toolkit. This is a catalog of recommended measurement protocols. It's not a data repository. The goal is to encourage researchers to consider using these recommended protocols to facilitate cross-study analysis and increase the impact of your individual studies. The funding, as I said earlier, comes from my institute, NHGRI, plus 7 other institutes across the NIH. The PI is Carol Hamilton, the Director of Bioinformatics at RTI International. And you can see the URL for the toolkit on your screen. So I encourage you to explore the toolkit, but I'll just give you a brief summary of the content. There's 746 protocols in the toolkit right now across 26 research domains. Some examples of research domains include demographics, For example, ethnicity, birthplace of grandparents, gender identity, education, marital status. There's substance use, social environments, which include perceived discrimination, healthy food environments, job strain. There's physical activity, including activity screeners and cardiovascular fitness, to name a few. There's protocols that span the life stages, so from reproductive health through pediatric development and geriatrics. And then there's a number of protocols that come from disease-focused domains, diabetes, cardiovascular disease, and oral health. And then there's also a number of special collections with additional funding from NIH institutes. They did a deeper dive into some of these domains, including mental health, substance use, sickle cell disease. And this week, there'll be a new set of measures in social determinants of health that will be added to the toolkit. So we like to say that these are consensus-based protocols that are added to the toolkit. There's a process that the Phoenix project uses. We have a steering committee to guide the overall effort. We have liaisons from all of the NIH institutes and centers. Each time we launch a new domain, they assemble an expert working group that does a sort of a landscape analysis of all the protocols that are currently in use. There's quite a bit of community outreach for get input on the draft protocols. And then they assemble expert review panels to go back and look at the toolkit content every couple of years to make sure it's relevant. And then these are just some of the criteria that are used for protocols to be included. They have to be broadly applicable, meaning that they're not relevant to just one sort of study or looking at just a nuanced research question. They would be low burden to the participants and investigators, validated and reproducible. The standard measurement protocol exists, and they're open source and non-proprietary. So I'll just give you one example. Here I'm using the search in the toolkit, and I decided to look up nicotine dependence. Then you'll see there's 36 protocols that are returned. It's a smart text search, so ideally and usually the first protocol that's returned would be the most relevant. So in this case, I do see a measure of cigarette nicotine dependence. The source is the Fagerstrom test. If I click on that protocol, you'd see a number of useful pieces of information. So there's specific language for how to administer the protocol. Again, considering that the idea is that this toolkit is useful for investigators that may not have experience in certain research domains. So they're trying to give them all the tools they need to deploy this protocol in their research study. You can actually see the text for the protocol. In this case, it's the 6 questions from the Fagerstrom Index. The Phoenix team generates variable IDs and data element dictionaries. You can also see there's a mapping to dbGaP if you click next. So in this case, variables within each protocol have been mapped to studies in dbGaP to identify other studies that have data on the same or comparable questions. This is quite a useful tool. Each Phoenix protocol has been mapped to a number of different vocabularies, resources, and ontologies, again, to facilitate data integration and harmony. And then lastly, I just wanted to close with something that's new in the toolkit. So as part of a trans NIH effort, Phoenix began cataloging the protocols that are currently in use for COVID-19 research. At last count, there were 45 protocols that were submitted to the NIH for posting. And those are organized on the Phoenix toolkit. If you click the orange banner, you'll get to this list. And each protocol is annotated to describe what the content is, what the modules are. So you can see some of them cover symptoms, knowledge and attitudes, adherence to mitigation behaviors like social distancing, social impacts, and economic impacts. And the idea and the hope is that the research community will come here and see which protocols are already in use and consider using them first before standing up a new protocol that then becomes more challenging to integrate over time. So here's just some statistics to give you a sense of which protocols and domains are the most accessed. So family history and mental health disorders is our #1 hit for our protocols, and the domain that's most frequently tapped into is substance abuse addiction. And there's more statistics available on the website. So I think that's it from me. I'm always happy to answer any questions that you have about Phoenix along the way once you take a look at it. Thank you very much.
Thank you. Yes, thank you, Anne. Very interesting for me, I have to say, that I wasn't aware of this toolkit before. How available is it?
The toolkit itself is it's free. It's online at Phoenix Toolkit. We've had over 1.5 million visitors. Lots of people are using it. There's a couple hundred publications now that reference using the toolkit. And they're working on adding translations for many of the protocols. There's Spanish and Chinese. I see someone else had, Dr. Mulder in the chat mentioned that they're using some of the Phoenix measures and have adapted some of the words or language for local context. So we're happy to see it being used more broadly and internationally.
While you were speaking, we did get some questions, comments that you probably didn't see, but Jeff is asking to what extent is that is the tool used globally versus in the US, in the US-based studies, and can there be an added culturally relevant measure to be able to look at tools that might be appropriate for a certain geographical site or social norm? Others are asking about harmonizing a minimal set of questions of, from out of all protocols on the same topic.
Yeah, so I'll answer that question first. There are, we try in Phoenix to minimize redundancy. So if they're going to pick, a measure of, this is a very simple example, but of ethnicity, there would only be one protocol to use to capture ethnicity. So we're trying to just highlight one protocol. And then as far as the core groups go, for example, the Mental Health Institute identified a set of core measures that they thought were most relevant for the mental health community, and those are organized in a collection on the toolkit. So yes, there are some core measures that are recommended. And as whether or not Phoenix is used in the US versus globally, I would say there are examples, like we mentioned from Dr. Mulder, that Phoenix is being used internationally. I think probably most likely that the measures are used in US-based studies, but part of that could just be the lack of dissemination of the resource. There are a few examples of measures that would be specific to the US, like using US Census data. But for the most part, I think many of the protocols would be globally relevant. Of course, you might have to tweak some of the words or language for the local context.
Okay, thank you, Aaron.
Yeah, thanks for having me.
Okay, thank you very much. Very interesting talk. Hakon, you take it from here.
Yeah, thank you all. We are fortunate to have three very impressive talks. It was a great extension of the strategy discussion from yesterday. Alicia gave us the genomics of the world impact, outstanding talk, basically emphasizing the key importance of moving out of European study populations that currently constitute 80% of essentially all data into a much more diversity populations, taking Africa as an example, Japan and other Asian populations. And on the Alzheimer front, the goal, impact, and visions we heard were very, very impressive and very much in alignment with the IHCC, where we have the ability together, I think, to have huge impact on this disease. We are not going to beat this disease except through collaboration and by generating sort of, innovative ecosystems, as George pointed out, that sort of are, comparable to cancer. That's what's really needed to tackle this disease. And the Phoenix Toolkit presentation by Aaron was similarly outstanding. This is a great resource, 746 protocols with 26 research domains, sort of going from demographic, social into reproductive health, pediatrics, and then disease-specific for multiple different diseases, and all mapped to dbGaP and other public databases. So outstanding resource.